You can spend years and years just working on one particular protein and trying to understand how it does one particular function, and we're trying to understand what the cell does, and that's the sum total of thousands and thousands of these proteins all acting in concert and regulating one another and responding to all sorts of consequences of their own action. Proteomics solves the problem of how you can identify all of the proteins that are involved in one of the many different activities of the cell. Our proteomics facility provides state-of-the-art mass spectrometry technology that can identify hundreds or even thousands of proteins in complex biological samples. Very large numbers of samples can be analysed using a range of high-throughput and robotic equipment. Essentially, a mass spectrometer is a very sophisticated balance or scale which can measure the weight of individual molecules or fragments of molecules. The weight of the molecules can be found by determining how long it takes for them to pass from one end of the machine to a detector at the other end. This provides us with extensive lists of all the proteins present in a sample, along with some information about how abundant they are. Proteomics technologies like these are used by a large number of groups in the GRE Centre. My research group uses mass spectrometry to identify proteins involved in copying the DNA inside cells, which must be done with extraordinary accuracy every time a cell wants to divide, as copying errors are a major cause of what goes wrong when cells become cancerous. Other groups use mass spectrometry to discover the proteins involved in the physical organisation of DNA in the cell nucleus, and proteins that regulate gene expression in response to different cell stresses. It's a really amazing technology, and it allows us to get vast amounts of data that just a few years ago would have been unimaginable. I think proteomics is going to be increasingly important to biological research in the future, and we're lucky in the GRE Centre to have this world-class facility so easily accessible to us.